Hello students, in this lecture video I'll be discussing about chapter 8, that is how do organisms reproduce. We will cover reproduction, introduction, and one type of reproduction here is covered, that is asexual reproduction. What is reproduction first of all? It is process by which any organism will produce its own kind. It is important for the continuity of species from one generation to another. Now, what is the basic event which is happening over here is, for the transmission of hereditary information, copying of DNA is very, very essential. So, if one cell has to divide into two, it has to contain equal amounts of DNA in it. So, copying of DNA becomes important during reproduction. Now, in case of sexual reproduction, two parents are involved and there are variations. So one thing is that two parents will give rise to an offspring and that offspring will contain both the types of genetic material. Another is in case of asexual reproduction also, sometimes variations are generated because copying of DNA is not 100% accurate. Sometimes some useful or maybe some harmful variations are introduced. So these variations become the basis for evolution. It helps organism to survive under unfavorable conditions. So let us first look at the differences between asexual reproduction and sexual reproduction. Asexual requires only single parent, whereas sexual reproduction requires two parents. There are no formation of reproductive cells or gametes in asexual reproduction, whereas in other, the fusion of gametes has to take place. Secondly, or you can say important point over here is, in asexual reproduction, the progeny is genetically identical to the parent. But in sexual reproduction, there are obviously variations. So let us look at the types of asexual reproduction. Firstly, fission. Fission means splitting. One type is binary fission. Binary means two, when the parent divides into two daughter cells, like in amoeba. Now, sometimes the cell is divided into two equal halves in one plane. In other times, in some other plane, like in Leishmania, we will see longitudinal binary fission happens. Coming to multiple fission, it means multiple organisms, or you can say multiple daughter cells will be formed. So, parent cell is splitting into many cells. Generally, this is happening under unfavorable conditions. When what happens, like only nuclear division take place and there is no cytoplasmic division. But under favorable conditions, these cells will be released. This slide shows you the variety of fission which can happen. First, A is showing binary fission, which is in amoeba. You can see clearly there is nuclear division, there is cytoplasmic division and leading to two daughter cells. Next, coming to this Leishmania, this is showing Leishmania, longitudinal binary fission like this, in this plane, division will happen. In case of Paramecium, again, transverse. So this is in one plane, binary fission. This is in longitudinal way. And this is in transverse plane. So these are different types of binary fission that can happen. Next, let's see the different type is fragmentation. See, binary fission was happening in unicellular organism, but fragmentation can happen in multicellular organism like Spirogyra. This is the algae which contains spiral chloroplast. We can see over here. It is multicellular. So fragments of single cells can occur and the single cell will give rise to a new organism. That is fragmentation. Point to remember over here is, in multicellular organisms, only those who have cellular level of organization can undergo fragmentation. Others cannot. Because if it is tissue, organ, like this, fragments cannot form the new organism. Next, coming to regeneration. In this, actually, there are basically organism breaks down into different pieces. And any piece can give rise to a new organism, like in planaria. But if we talk about lizard, it is regenerating its tail, regenerate, reform, okay, forming again. In that case, it is a type of development rather than reproduction. Next, budding. You have done this in lower classes also. Here in yeast, it is being shown that a bud or outgrowth will be formed, and later on, when it matures, it will just be released and daughter yeast will be formed. Next is spore formation, in which spores are being formed. Like in fungus, there is a sac in which these small single celled spores are formed, which will germinate into a completely new individual. So, this one spore will give rise to this whole fungus. Next is vegetative propagation. Vegetative propagation generally happens in plants where the vegetative parts like stem, roots, leaves, they are taken like cutting, or you can say layering. Like I want to grow a rose plant and maybe in my neighborhood it is growing, so I'll take a cutting and I'll grow it. So that is vegetative propagation. What are its advantages? Firstly, uh, plants can bear the fruits without, you can say, then those which are producing from seeds. Without that, also they can produce. Then it enables the different plants like banana and all, which have lost the capacity to produce seeds, to propagate. And also, it maintains the genetic similarity, it preserves the genetic material in plants. Like one example is of bryophyllum, wherein at the notch you can see small buds are arising. When these will fall off in the ground, they will give rise to entirely new plant. But there are disadvantages also. Disadvantages: plants will lose their genetic variation because similar genetic material is there. And sometimes overcrowding can take place because of the habitat, you can say resources are limited. Lastly, coming to tissue culture, which is in lab how we can grow plants. The cells from the tip of the plant, that is dividing part of the plant, are taken. In artificial medium, they are put and small undifferentiated mass of cells will grow, called as callus. Further from which plantlets will be formed and then we can grow it into the soil. Now, this will actually help us to obtain disease-free plants. See, in natural environment, a plant can get disease, but in lab conditions, we can avoid this. So generally for ornamental plants, this tissue culture has been used.